We start again. So what we did is the tissue pulverization. I was taking a look of the samples. They are quite good, still a bit of big chunks inside, but this is not a problem. It's not gonna interfere with the process because we will still have some DNA inside. So we did step 1 and we did step 2 with the lysis. So the lysis buffer plus the proteinase K. They worked for 2 hours at 50 degrees, so they try to give us the DNA, make available the DNA from the cells. And now we are going to proceed with the last step, that if you are fast in 1 hour, it will be done. That is DNA isolation. Okay, now the chain of cold is broken, so we don't have to work with liquid nitrogen anymore. Okay, now I don't know if you can see the settings from here, but this is what we have to do. So first, in front of you, you will have this kind of Lego shaped stuff. You have to take one, all right? In front of you, you have an empty Falcon tube. You have to open it. It's there, Elena. And you have it here, so yes. And you have some tubes there, the empty one. Okay. I'm gonna show you here maybe on the screen so you can see what I'm doing. I'm gonna put the tube here, and then you have, this is the genomic tips. It is a tip with a membrane that is gonna first retain the DNA and then release it, depending by the buffer that we are using on it. And you have this small yellow circle. Do you have it? Yeah. In front of you? I mean, bench number 4, you don't have to open the tips. I'm sorry, but yeah. Yes, so what I do now, I open the genomic tip with gloves, please. I take it from the top. I put the yellow ring here. I put it inside and I let it go in the Falcon tube. And then it stays like this. In there? Okay, yes, perfect. In the empty one. In the empty one. Okay, you open the genomic tip, you place it within the yellow circle, and then you place it within the Falcon tube. And then what we are doing, you have a buffer that is called the QBT in front of you, 1 ml. You're going to take 1 ml of the buffer and you're going to place it within the column, within the filter, the tip, without touching the membrane, the white membrane, okay? So what we're going to do, you're going to take 1 microliter QBT of buffer QBT. I'm going to take 1 ml. You should have enough inside. Sorry? Equilibrate means bring to work. Exactly, activate the membrane, because the membrane, when it's dry, is not active. It will not retain the DNA. And then you put it inside. No, you touch as less as you can. Yeah, the filter tip. But not the membrane, the white membrane. Yeah, exactly. Yeah. The membrane. And then you can observe that it will start to drop. You will have some drops falling down. And then let me know if you see the drops. We are DNA isolation. Step A. 1 ml. Sorry? No. First, we need to activate the membrane, because the filter tips are dry and they can stay dry up to 5 years. But you need to activate them. So now with this buffer, we activate it. So when we place the sample is going to be retained. The DNA is going to be retained first in the membrane, and then we wash with 1 buffer and it's going to go down. All right? And if it's too viscous with too much DNA and fragments, as you can see, there are some fragments still. It can take a lot of time to pass through the filter. So if you put 2 ml directly, it's gonna go down faster. Okay? Okay. So is the next step clear? You take the DNA inside, you put it on the tips, and you wait that it drops, okay? And if it's not dropping too fast, I will come and explain you how to make it. No, not yet. It's fine like this, because if you centrifuge, you will lose everything. If you do it like this, the biggest chunks of tissue will go down, but DNA still suspended. If you centrifuge, it's gonna go everything on the bottom, and then you're gonna lose everything. Only the buffer will remain.
Depends on what.
Yeah. If you take some of the tissue, it's not a problem because the filter is there for this reason. Because now we have time for questions. Yes, even about the previous step.
How expensive is this PhD?
The tips are not expensive. The buffers, the set of the buffers, it costs around €200. And you have, oh, this is a good, there are different sizes. And even for the tips, these are the smallest one, and they're not, maybe in the same range, 200 to 300. Yeah. So with 500, you can have, I believe, 20 samples, 25 samples. Yeah, it's a lot. You will always have leftover from this kind of kits. It depends how many species you are assembling. You will assemble, but you always have leftover even with the smallest one. Need to use a different set of buffer. I mean, when you buy the buffer set that is €200, there are all of the buffers for bacteria, blood, tissue sample, and tissue sample include plants and fungi. So this protocol you can use with all of the plants, and usually it's working. This is what we routinely do at University of Lausanne. But the only difference that we have is that for the pulverization, we have a machine that do this. So you have no loss of, you have no condensation of water. Somebody else was saying before, there is a lot of water. There is a machine that brings the tubes to minus 180 with the magnetic beads inside, and then it shakes super fast for 30 seconds, and then you have your powder. And you add the reagents inside. You leave the beads inside. It's cryomill. Tissuelizer it's a bit of an old technique now. Okay, now for the one that are done, you can see that you can start with the buffer QC. It's a wash. So now you're going to wash everything but the DNA from the membrane. All right? Okay, you can start. For the one that are done, you can start. Yeah. Elena, you can start the buffer QC. Yeah. You have two sets, two aliquots of buffer QC. Yes, Alice, please. Yes. Yeah, it is. So 1 milliliter. As I said before, you have to play with gravity. If you put 2 milliliters, the pressure is going to be higher. So I suggest for the first wash, you put 2 ml in a row. Okay? And then when it's half empty, you add the third one. All right? Yes, there are two aliquots. Same, yes. You put 2000 first, and then when it's half empty, you add the last one. QC1 and QC2 are exactly the same, so you can go with one or the other. Perfect. I mean, when you will be in the lab, you can wait up to 3-4 hours. Yes. You don't have... When you're working, you just leave it and then you wait that it goes down. If you avoid to use it, it's better. But if you can use it with 5 drops per minute, it means you just do really slow. You have to find your rhythm. Yeah. Just wait 1 minute, and if it doesn't go, you can use it. In front of you of isopropanol like this. All right, do you all have it?
Yeah.
Yes, perfect. And you can... Oh no, we will add it. No, nothing, it's good. Just wait. So basically now what is happening, the DNA is going to get dissolved in the buffer and is going to get down, if there is any. I hope so. Physical properties? Yes, yes. Huge chunks of DNA can pass through the membrane without being destroyed. I mean, once we did for an insect and we arrived to chromosome level only with hi-fi data without using IC for the genome assembly. So if you could do it well, it can yield really good results. Usually we have good results. And the kit is not expensive. It's one of the cheapest solution that you can do. Yeah. But when we will write the report, I will send all of the links for the kits for everything. So now the DNA is collecting down. Now you can, when it's done, you can trash the genomic tip. But now the buffer has different properties, and instead of keeping in the membrane, it's going to release it. Yeah. pH, electrical charge, and the solvent. There are soaps inside or no soaps, so it depends. Yeah, it's the chemical properties of the buffer. This is a 50 ml tube centrifugation. You have to centrifuge your Falcon tube of 50 ml without putting into another things. It's written in the QIAGEN protocol. In the QIAGEN protocol.